My name is Yakobson. I actually was born in Riga and graduated from Latvian University, then went to Moscow for my PhD, then came to Israel to do another PhD, and then was in London, advanced studies, California 5 years, came back to Israel, twice visited the States for 2 years, and settled in Israel for altogether 40 years, and now came here to the last part of my curriculum vitae. I was very warmly accepted by Ingrid Smuznieks, gave me the title of professor, and now I'm establishing a genomic unit in here together with Professor Trifonov and Professor Thomas from Munich. We just had a good meeting to establish our unit. What is advanced genomics? It's something very new. A few years ago, when 7 years ago, first DNA was sequenced, first genome. And they took $20 billion and many people to get through it. Now there is a machine smaller than this desktop which does it for $1,000, and in one day you can have individual DNA sequence, 3 billion of letters. Why decided to talk here in this conference? Because Latvian jury, about 10,000 strong, is an excellent model to explore genomics as of a compact, relatively isolated community, its structure and its characteristics. With the advent of genomic high-throughput sequencing, it became possible to reveal full genomic digital signature of individual Homo sapiens in one day at a price under $1,000. Newly developed affordable desktop nanopore DNA sequencers are supporting this technological revolution. It's a real game changer for personal genomics and ethnic genomics. Jewish people, on the other hand, are particularly suited for this line of research, representing an isolated community for about 3,500 years with a very well documented biblical and genealogical record. I'm exploring genetic and historic aspects of Jewish people for the last 27 years. We have reference in PubMed, and we will discuss how this digital DNA revolution will be in this conference, how it applies to all research. So two new developments, this next generation sequencing, single DNA molecule sequencing, and their impact on it, I already mentioned. The so-called copy number variation, it's anatomy of the genome, so not everything is linear. There are spaces which repeat itself 4, 8 times sometimes, and there are deletions. So it gives an added complexity. There is at the Harvard University, Personal Genome Organization, headed by Professor Church, and they already collected about 2,000 people. They sequenced more than 200, and this is a major source and inspiration for my research too. So somebody who is interested in the technology, I can give these links. both on YouTube. So if a whole genome takes about 1 day and $1,000, so the specific regions of DNA could be sequenced much less expensive, and more people could be, in 1 day analyzed and compared. Ethnicity is also a new concept in genetics. There is a guy, Joel Saldacora, who published a book talking about 200 Jewish diseases, which are in Jews are more frequent or less frequent than general population, or different at all. And Ashkenazi Jews differ from non-Ashkenazi Jews. How I come involved, I studied predisposition to melanoma, and I saw that Jews, Ashkenazi Jews have different location for melanoma than non-Ashkenazi Jews, and it was a major new thing, which made me to think about ethnic genomics and Jewish genomics and ethnic genomics. So what are the major changes? The genomic deletions, which are more frequent, rearrangements, amplifications. And it was shown for malignant melanoma, breast cancer, Parkinson's disease, Alzheimer's disease, and many other disorders. So what people do, and I'm doing, is establishing databases for mutation detection, disease prevention, molecular epidemiology, biotechnology, drug development, biobanking, and national germ DNA repositories. And this is important because ethnic identities, they differ in their sensitivity to drugs, and it's very practical. Let's say there is a blood pressure drug in the States, very popular, works only practically for Afro-Americans, doesn't work for European Caucasians, and this is